So I'll be talking about the AI-driven spatial transcriptomics for predictive drug responses in tumor microenvironment. My name is Kayode Raheem, so I'm from University of Nebraska Medical Center. So firstly, why are we trying to work on spatial transcriptome? What are the challenges? So firstly, the challenge is more on how tumors, we should know that there are mosaic cells of cell types and cell state and microenvironment. So these are kind of spatial heterogeneity that drive drug divergences. That is, it makes drug responses differs across different modalities in terms of the bulk and traces single cells assay that couldn't resolve. So that's major challenge of trying to face. So what are the opportunities we can able to address this? And so in the recent advancement, like in the spatial transcriptome platform, so we have a lot of platforms that are able to produce a lot of vital information in the sense of their gene expressions and even the analysis, able to understand the cell-to-cell interactions, tissues that help us to understand the therapeutic shapes at this point of the molecular level. So what are the recent gaps in this field? Precisely the spatial transcriptome in the sense of drug therapeutic target identifications. So most of these computational out there tools, they majorly focus on what? On a single modality. That is, they focus on single input, like the gene expression only, which they kind of ignore the histology information at the proximity of where the cells are located in the tumor regions. So this idea of how we can be able to utilize all the three modalities to be able to transfer the knowledge from the pharmacogenomic cell lines to a patient tissue due to that domain shift. So these are the major kind of gap we're trying to bridge the gap in terms of our computational tools we are trying to develop to address these significant questions. So what's our innovation with the tools we are trying to develop? So the tools we just develop kind of leverage on these transfer learning skills. So like we try to transfer the knowledge from the bulk RNA level to infer the information on the spatial data. So in the sense that we train our label cell lines using these adversarial domain adaptations. In sense, we kind of align the tissue embedding and assign the prediction properties in sense of the affinities and the system label per cells. So this kind of tool is a kind of incorporative modality, and that it helps to integrate the gene expressions, the spatial coordinate, the histology in kind of a unified matrix. So why this is that to be able to kind of infer more biological information that was not able to done when considering the single modality. So in this architecture, kind of introduce a more advanced AI model that we introduce a graph neural network because it can able to infer more of these spatial information from the cells, spatial cell precisely. So it can able to improve the prediction mechanisms in the drug therapy target we are trying to address. And also, in terms of alignment of these two source domain that's upon this target and target, so we kind of introduce the word adversarial weighting to do that. So the impact of this project was to expose what the therapeutic resistance and the sensitive cells within what that being invisible when considering the single modality method. So this part was kind of being addressed with this computational tool we are trying to build in this phase of that. So this kind of give a pictorial view of our entire workflow we trying to implement. So we can see we have the source domain to target domain, and our target domains are spatial cells, right? So we're trying to infer some drug responses that are not being available when using the spatial cells. So our tools can able to predict responses to more of these spatial cells in that regard. All right. So what are the kind of method we try to implement to do this particular workflow framework we did? So the spatial cells, like we kind of considered what the histology images, and this workflow has been done using the ability of the Nextflow techniques. It's a kind of a standardized pipeline we developed to able to build this pipeline to predict your response based on the spatial cells. As we can see, the histology images and the spatial expression and the coordinate were being considered in this algorithm where we kind of concatenate the expression we get from the target and the source, where we try to do a kind of transfer learning, and that led us to do the domain adaptation method here, where the drug tutor will do what to predict responses based on how the knowledge is gotten from the source domain, and the source domain is actually coming from the pharmacogenomics profiler scale we try to implement. So this led to prediction of these spatial cells. How some of these spatial cells are responding to different individual cells, individual drug we try to infer. So the training method was based on using some advanced techniques, and that's the mean distribution adaptation method. So we kind of give the responses based on how each drugs are actually responding to the spatial cell, precisely the locations. So that was done in the kind of workflow we see here. And it's actually advanced a kind of a convolutional network where we kind of extract the meaningful features from histology. We kind of concatenate with the gene expression profile to get the biological information to that. So what our framework was able to do, checking the matrix against, or we kind of checked it against some existing tools, we found that as you see on this lefty table, you see lots of drugs. We try to see how each of these spatial cells are responding to them and based on their metrics. So looking across, we see that the method we are trying to kind of develop have the kind of more predictive accuracy, and it has a higher more accuracy like in each drugs to see to that. So this kind of give a brief comparison with the existing method. So we found that you see our method is one in purple. It kind of outperforms the other existing method with a little bit percentage average to that. So with that, we did that across different drugs, and we see the performances in each drug. So we can see that different drugs have different performance based on the classes of their data we use to train our model. So our model was able to work fine to predict responses to especially to what the cell levels of these cells across the domain. Okay, so I try to show you kind of how we now try to implement these tools to a real door data as a lung cancer cell lines, like the spatial cell, to see how the tool was able to predict response to this particular drug. If we're seeing, we can see this is showing precisely the location of each cells at the tumor region. So the spatial visualization of the cells of each cell, you can see numbers which are sensitive and which are resistant to that. So we also see their level of interaction, the cell-cell communication, how are these cell types are also having effects based on their responses across in the cell regions of the tumor, precisely lung cancer. So we can see these are different field of view in different locations. We see our rate of these cell types, precisely B cells, the NK cell, how are they responding, which of them are sensitive, which are not resistant to the drug we're trying to show. So we also do a kind of summarization over all the entire field of view of the entire section of the lung cancer. We see how each of these drugs are actually resistant to this particular eposin cells. So this map shows us this particular region of which cell types are more sensitive and more resistant, which are major focus of this our work we try to implement. Yeah. So thank you for listening. So if you have any question, just let me know. Thank you.